We are back with DigiPath Digest, fresh from PathVisions. In this episode, you're going to get some PathVision app updates. Then we're going to talk about advances in AI for breast cancer diagnostics and how digital pathology is making its way outside of pathology departments into primary care and global health initiatives. So let's dive into it.
Learn about the newest digital pathology trends in science and industry, meet the most interesting people in the niche, and gain insights relevant to your own projects. Here is where pathology meets computer science. You are listening to the Digital Pathology Podcast with your host, Dr. Alexandra Zhurov.
Good morning, my digital pathology trailblazers. Let me know that you're here. I see you are coming in. When you are here, let me know where you are tuning in from. I have coffee ready and I have a few updates for you. Let's start with PathVisions. So, PathVisions was this week. Okay, we have people from Poland and Lebanon. Amazing. Welcome, international trailblazers. PathVisions was in Orlando. What's PathVisions? PathVisions is the annual conference of the Digital Pathology Association, the DPA. I was there, and this was amazing. I was there to support my sponsors, two main sponsors this time. That was Apredia and MUSE. Because of them, I went there and benefited from the whole conference and met many amazing people. So let's talk about the trends I've noticed there. There are three main trends. And one we have been seeing in our DigiPath Digest already. I've been talking about it, I think, last week or a couple of weeks ago. The first trend is integration of narrow AI into the diagnostic workflows. So the digital pathology journey is long, and there was a talk by Dr. Anil Parwani who compared it to a train ride, and it has many stations. It usually starts with what scanner you're going to choose, goes all the way to AI, diagnostic AI. And I see some more people joining, so feel free and let me know where you are tuning in from. We have from scanner to AI. It's a whole journey, and there are different stops on this digital pathology adoption train. For those already on this journey, the stop where they have to figure out how to do it and then keep going is to integrate the AI applications for diagnostics. That will be an app to count Chi-67. There are different apps available for diagnostic scorings. That's what's happening now. And we've been seeing that in the literature where people are publishing, okay, we use this algorithm to quantify PDL1, to score colorectal cancer, to count Chi-67, and it shows up across different journals. People are doing this work, figuring out, okay, is it as good as we are? They're figuring out, okay, is it as good as a pathologist? Does it correlate? Can we use it? This is the hurdle that the people who have been on this journey for quite some time need to overcome right now. The second trend that I've seen was integration of less narrow AI. I don't call it general AI because to me, when you talk about artificial intelligence in a more conceptual way, a general AI would be as good as a human. And there is nothing like that out there, but there is less narrow AI that can do text and images. So obviously foundation models we've been talking about, and the chatbots, meaning the large language models and the integration of multimodality. So I see the integration of this into slide viewing platforms. So there are different partnerships, for example, PathPresenter, which is a slide viewing and image management software, partnered with Page AI, not only to deploy the now AI algorithms that Page AI has developed, but also to integrate the new wave of AI into where pathologists are already diagnosing, into where pathologists are already looking at images, which is fantastic, because the previous trend, the integration of now AI, has the problem of being able to access different apps from different vendors, but not in the same place where you're viewing the slides. It may seem like a first world problem. But if you're going fast through your cases and every second counts, you don't want to be clicking between windows. Now the vendors are figuring out how do we partner and how do we make it seamless for the pathologist so they can deliver that diagnosis faster for the patient. There is a third trend, glassless pathology. If you've been listening to me for the last month and a half, the digital pathology place sponsor MUSE is in that category, glassless pathology. They have a device that images direct from fresh tissue. There's no need to do formalin fixation, paraffin embedding. We don't need to do the normal glass slide workflow. We can just image tissue. And MUSE stands for microscopy with UV surface excitation. So you excite the surface, you image it, and you have an image that is diagnostically as informative as an H&E image, and it actually looks very much like an H&E image. That is the new wave. I was talking to Giovanni Lujan, and we called it the new frontier. 10 years ago, by the way, Digital Pathology Association is celebrating the 15th anniversary. They had special cookies for that. I made a short, so check it out on LinkedIn. The next frontier is integration of those new developments in AI into where pathologists are actually looking at slides, plus glassless pathology. So I'm super excited about that. It's the 15th anniversary of Digital Pathology Association, right? Some of the people who were there, not only vendors, also the participants were there every single time. It was my second time. I remember the first time I came with a poster about digital pathology in drug development. People had no idea what to do with it. The audience that comes there is mostly MD pathologists and the digital pathology professionals from the clinical space, a little less from drug development. So people didn't really know, like, how do you use it in drug development? You use it for clinical trials. They had no idea that there is something like preclinical work for drug development. My last conference was in 2019. Fast forward 5 years, people are a lot more informed about the cross interactions of different disciplines where digital pathology can be deployed. Shout out to the digital pathology crowd for advancing so rapidly. And another thing that was interesting, there's a lot more collaboration with radiologists. So there was a talk under the umbrella of Society for Imaging Informatics in Medicine, SIM. And most of the members of SIM are radiologists, but now they're inviting more and more pathologists, which is fantastic. While I am talking about the cross population of different disciplines in digital pathology, let us go to our papers today. First paper is AI-supported digital microscopy diagnostics in primary healthcare laboratories. I want to highlight primary healthcare laboratories. If you have any questions regarding the papers or anything, put them in the chat because then I can answer them. I love answering your questions. Primary healthcare laboratories. This is a protocol. It's not a full publication. I was reading this abstract and I'm like, where are the results? It's a protocol, so there are no results yet. And I'm excited because it's a primary healthcare setting. Basically, digital microscopy, it was called digital microscopy because it's actually microscopy. You can use it not only for pathology. Pathology is the science of disease. Any disease for me is pathology. But in the more classical way, it's more like cancer diagnostics. Primary healthcare laboratories. I like it because it's a One Health approach so One Health is where you combine human medicine, veterinary medicine, and environmental sciences to improve healthcare. I am a veterinary health professional, so that's my part of One Health. I gave a talk one time in Africa about that. These are research protocols, impact factor 1.4, basically telling us what study they're going to be doing. This is a Department of Global Public Health in Sweden, and we have Institute for Molecular Medicine in Finland. They're combining digital microscopy and AI, and it is being implemented in advanced laboratory settings. Yes, it's pretty advanced, pretty specialized. It could be especially advantageous in primary healthcare settings. You could use it for different things, looking at fresh tissue in the future, looking at blood. This would improve access to diagnostics via automation and lead to a decreased need for experts on site. Yes, decreased need for experts on site. There are not enough experts to have them every time on site. This is a scoping review, a specific type of publication where you map different things, how to do it. So they mapped peer-reviewed studies on AI-supported digital microscopy in primary health care laboratories. And the method was a systematic search. Database is PubMed, Web of Science, Embase, and IEEE. This publication is now this month, last month probably, November 1st, it was published. So I am actually surprised when I look at these new publications, there are no AI literature research tools implemented yet. I had to make a video on them, but it's still PubMed, Web of Science, Embase, and there are different ones, right? But no AI tools, just the databases. They will get there. They only use peer-reviewed articles in English. No limit of publication year will be applied. Studies that have applied AI-supported digital microscopy with the aim of achieving a diagnosis. So this is important. It's going to be for diagnosis on the subject level. These studies must have been performed in primary healthcare laboratories, and the criteria was they did not have a pathologist on site, and they were using simple sample preparation. Simple sample preparation is going to be cytology, smears, blood smears. It can be fecal smears. There is a super cool publication that also talks about One Health that we're going to discuss in a second. And the question is, will there be a recording available? Yes, the recordings are going to be on YouTube and whichever platform you're watching it on. But also, if you're on my mailing list, you're going to get the recording to your inbox. So if you're not yet, then go to digitalpathologyplace.com and download the book. Then you're on my list and you're going to get the recording every time we have this session. They didn't have a pathologist on site. That was the requirement. I am very curious what they're going to come up with. So they anticipate to identify the diseases where these novel methods could be advantageous. These were the simple sample preparation methods, smears, cytology, but there is going to be the new wave of imaging of tissue, and that is going to be the direct to digital from tissue. This is coming, so whenever it's out, I want to be the first to let you know. Digital microscopy in primary healthcare settings. Last month was Breast Cancer Awareness Month, so we do have a lot of breast cancer papers from October in histopathology, impact factor 3.9, and where are we talking about implementing narrow AI again? So Chi-67 evaluation using deep learning model-assisted digital image analysis in breast cancer. This is a publication from Japan. What did they do? They checked if Chi-67 by AI is good enough. And they said yes. They checked the efficacy of AI-assisted Chi-67 digital image analysis in invasive breast carcinoma. And I'm going to tell you why I want to talk about it. In 1 second, they did quantitative assessment of AI model performance. Standard thing, you have a model, you check if it's good enough, and then you decide what to do with it. And we have seen that most of those papers says, oh, it was good enough, but we are not ready to do anything with it. For this particular study, we had 494 cases of Chi-67 slide images from invasive breast carcinoma and needle biopsies. It wasn't resections, like the initial diagnostic settings where they take a biopsy from your tumor. They had 2 steps. They developed an image analysis algorithm, deep learning model from scratch. And they did it using Halo. Halo involvement here is the reason why I wanted to bring in this paper, because it's something that is commercially available. You can get this software and you develop this algorithm on your own as well. You don't have to have any fancy integration. You don't have to buy the algorithm. You can just develop it on your samples. You're going to be sure you do your own validation. You're going to be sure it's going to work on your lab because you did it, your team did it. And that is the advantage of doing it with commercially available software. There are disadvantages and advantages to everything. If you want to do like a homegrown on your own, there are platforms that are going to help you do it. And Halo is one of them. They divided it in 2 steps. First, tissue classifier. So they found the tissue with the classifier, and then nuclear detection model, because for Ki-67 evaluation, you need to have it in the tumor, detect all nuclei, because you need to count the positive for Ki-67, which is a proliferation marker, and the negative, and then calculate an index. They had almost 32,000 annotations, which is annotating just single cells, in 300 Ki-67 digital slide images. And it was compared with the annotation of ground truth at the pixel level. They probably did semantic segmentation rather than object detection. It was performed on 194 luminal type cases, and the correlation with manual counting and clinical outcome were investigated to confirm the accuracy and prognostic potential of this image analysis. They liked it. The performance was excellent. The numbers were precision 0.85, recall 0.88 almost, and F1 score 0.86. High Ki-67 index cutoff, 20%, showed significantly worse recurrence-free survival and breast cancer specific survivals. Above this cutoff, the survival was worse. I like it. This plays into our trend of validation and using narrow AI. We have a publication from Radiology about a multi-label artificial intelligence approach for improving breast cancer detection with mammographic images. What is multi-label? Usually when you do classical supervised deep learning, you assign just one label to a specific class. But here they assign multiple labels, and that helped them actually. This is from in vivo, impact factor 1.8, and the group is from Republic of Korea, and these are radiation oncology researchers and also AI researchers. So what did they do? In this study, they aimed to develop a deep learning-based artificial intelligence model that predicts the malignancy of mammographic lesions and reduces unnecessary biopsies in patients with breast cancers. How can you be certain that this is good enough in terms of regulatory? Does it need to be an FDA cleared product if you actually want to use it as such screening tool? Regardless of the implementation, let's look at the result of the research. Their deep learning based AI was used to predict whether lesions in mammographic images are malignant. So if it would be not multi-label, but one label, you would have the mammography image and then the label malignant or benign. That would be the pairing of labels and the training of the model. But here they had this multi-label training similar to the diagnostic process of a radiologist. A radiologist then does not just say, your cancer is malignant. That's it. No, they look how big it is, where it is it in the lymph nodes? And I see more people joining, so let me know where you're tuning in from. Say hi in the chat and let me know. The doctor is not just going to say malignant, benign, goodbye. They will give you a full report. They assign it multiple labels, different types and different things. Let's look at the labels used in this particular research setting. They used curated breast imaging subset of digital database for screening mammography. And the data sets includes annotations for mass lesion. And the multi-label classification approach enabled the model to recognize the malignancy. So they had the information, whether it's malignant or benign, and lesion attributes. In the multi-label classification model, trained on both lesion shape and margin, here are the lesions attributes they used, and it was compared with models trained solely on malignancy. We added this additional information. What is the shape of the lesion? What is the margin of the lesion? And the information about malignancy, we had a better predictor. By considering margin and shape, the model assigned higher importance to border areas and analyzed pixels more uniformly when classifying malignant lesions. This approach showed improved diagnostic accuracy, particularly in challenging cases. The challenging cases, such as for every disease, there is a specific diagnostic system or diagnostic guidelines. The American College of Radiology breast imaging reporting and data system categories 3 and 4 are the ones that are challenging. The breast density exceeds 50% there. And the conclusion is Potential of AI is improving the diagnosis of breast cancer. I always laugh when that's the conclusion of a paper. The potential. Congratulations to everybody who is waking up or staying up late. What I wanted to say about people waking up at 6 a.m. and me hosting this at 6 a.m. One day, I think it was the third day of PathVisions, and I meet a friend whom I already talked to several times, and she looks at me and I'm like, "You look tired." I'm like, "Okay, I guess I am tired. I look at myself in this camera with you and I look tired, but that's okay. We show up tired and we do it anyway." By the way, have you noticed that my glasses don't glare anymore? You don't see everything I'm looking at on my screen because I got specific glare-free, what are they called? Anti-glare lenses specifically for streaming live so that you don't have to look at everything that I'm looking. The next step, I need to check the Meta glasses with camera. A friend, Nick Best from Pathology News, had them on at Pathology Visions. And I didn't believe it because I saw it on a billboard and I'm like, no way, they already launched and people can buy it. Yeah, you can buy it and they have cameras. And virtual reality is the next thing coming up in digital pathology. Let's go back to our papers. If you have any questions or just joined, let me know where you're joining from and your question. This is another paper and it has the One Health component. But actually, it's a study protocol. So they leave me excited, but then I'm like, where are the results? Well, there are no results because they didn't do the study yet. What they're going to do ties into the first publication that we discussed, the one about using digital microscopy in primary health. Here, they want a comprehensive evaluation of an artificial intelligence-based digital pathology to monitor large-scale deworming programs against soil-transmitted helminths. This is a study protocol published in PLOS ONE. I like PLOS ONE because it's an open source journal and the papers are published under Creative Commons license. So you can just download them. When I wanted to publish at some point under a Creative Commons license or open source science, I realized that you actually have to pay to publish it. You probably have to pay in every journal, but that was my first attempt in publishing something. I'm writing something and you charge me to publish it? Yes, they do. I was naive at that time. That was maybe 15 years ago. What about those deworming programs? This is the group Public Health from Belgium. We have group from Sweden, and we have groups from Ethiopia and Uganda. I visited Ethiopia, and that was where I gave the talk about One Health. So super cool to see this paper. What are they gonna do? They are gonna evaluate something that was evaluated manually or visually through the microscope with AIs. Manual screenings of cat-to-cat thick stool smear remains the current standard to monitor the impact of large-scale deworming programs against soil-transmitted helminths. So what is this? This is a way of preparing stool samples to see the parasite eggs. If you can see those eggs with your microscope, then you can develop AI to see them as well. They designed an artificial intelligence digital pathology system, AIDP, for digital image capture and analysis of those smears. I'm interested how are they capturing the images? It's probably not going to be scanned. It's probably going to be capture of digital images with a camera. This is the way that this is being done in developing countries. We recently looked at the publication from Pakistan. Once this is out, because this is a protocol, we're going to learn that. They want a comprehensive evaluation of this technology as a cost-efficient and end-to-end diagnostic to inform the soil transmitted helminth control programs against the target product profiles of the WHO. This is the next step of their validations. What are these target product profiles? When you have something to use against a disease, a medicine or something, it has a profile, what it works for, and this is the target product profiles. We're going to be checking, does it actually work for the parasites that it's supposed to work? Our methods are the comprehensive evaluation based on diagnostic performance, repeatability and reproducibility, time to result, super crucial, time to result, cost efficiency, and to inform large-scale deworming programs, and usability in both lab and field settings. Field settings, in this particular case, this is super important because maybe not everybody has access to a lab. It needs to be doable in the field. I'm super curious. Maybe they're going to figure out something like how to do it on the smartphone or something. Those researchers are creative. They will conduct in 2 soil-transmitted helminth endemic countries with national deworming programs. That's going to be Ethiopia and Uganda in school-aged children only. This is an official clinical trial. Trial registration was registered in September 29th, 2023, year and a month. Clinicaltrials.gov. I am super excited about seeing the results of this particular trial. These are our publications for today. I'm going to give you a little bit more update coming out on the content side from PathVisions. I wanted to give the shout out to the vendors who go there. It's intense. It's like a boot camp because you come in, set up the booth. Those people have been there for 15 years, joining conferences and trying to convince people to go digital with whichever product, right? It doesn't matter where you enter on this train ride of digitization. Maybe you're gonna enter in the future where the previous stations are like gonna be abandoned already. But wherever you're gonna enter it, there is gonna be somebody enabling you with a commercially available device. These are the vendors. Shout out to them. I had the honor to interview Jun Fukuoka, president of the Asian Society of Digital Pathology. This is a recently founded society, and he gave me an overview why we need a specific Asian one, how diverse the Asian society is. He even showed me a super cool AI tool. So I was interviewing him. He's Japanese. He speaks great English, perfect English, but he had this AI tool on his tablet that was transcribing me live and then translating on the other side. He had this, and he says, this is a mandatory tool for all the members when we meet on our society meetings to use, because there's no way you're going to have the same level of English and one language to communicate at the same level. So we need to leverage AI and the translation to do it. I talked to him, and it's amazing what's happening in Asia and how diverse this is. You will have the adoption of digital pathology from 50% in the Arab Peninsula countries to less than 1% or 0% in some countries. For comparison, in the US, people say between 5 to 10%. I do not have a publication to cite that actually counted this, but this is more or less the number. They say less than 10% in the US, where a lot of development is happening. Then I also met with 2 other educators in the field of digital pathology. One was Nick Best from Pathology News. He approaches digital pathology from, okay, what tools are there? Can you compare them? What is the best informed decision you can reach on your own before you actually reach out to your preferred vendors? They have this special technology buyer's guide where you can compare different solutions. I talked with him and Imogen Fit. She is from Signify Research. And she has this monthly recap. Oh, what's up in digital pathology space? Who partnered with him from the business perspective? What's going on? What are the trends? So we had the podcast together. Then there's another podcast coming out with Ella Kwam. She is in the content creation committee of the Digital Pathology Association. They had a podcast set up there as well. I was a guest, so stay tuned for that. There's going to be a vlog on YouTube. There are shorts on different platforms on YouTube as well. And by the way, an interesting thing I've noticed, there's a lot of trailblazers coming back to the YouTube channel, watching videos over and over again. Like 70% of you are coming back, but are not subscribed. So please hit the subscribe button. It's free. And if you're coming back over and over again, then just hit the subscribe button. I would love that. It's not only going to help you stay on top of the things, but it will give a signal to the algorithms to show it to more people who might be interested in digital pathology. And this is the point of this podcast. So thank you for that. So thank you so much for subscribing, and I talk to you in the next episode.